I think 15 years ago, someone asked me, well, what's the outlook? And I didn't have any future wisdom about these new therapeutics in the pipeline, and it didn't look that grim. Right now, it looks fantastic.
Hello, and welcome to this special series from Karger Publishers and the POLG Foundation. I'm Will Mountford, and over the next 3 episodes, we will hear from scientists discovering, clinicians treating, and patients living with POLG-related disorders, one of the many mitochondrial diseases. The story of the mitochondria is an interesting one. The origin of the mitochondria is a fascinating tale of symbiosis. Although these organelles are essential for providing energy and metabolic functions in all eukaryotic cells, they likely began as independent organisms. The endosymbiotic theory posits that mitochondria were once free-living prokaryotes engulfed by a larger host cell. Over time, this relationship became permanent and mutually beneficial. This merger provided the host with a powerful new energy source, but also introduced the mitochondria's own independent DNA. This separate genetic legacy is the reason why mutations in mitochondrial DNA can lead to a range of devastating, heritable diseases. In this series of videos, we will focus on the POLG gene and the gene product. POLG is a gene that provides the instructions for making the catalytic subunit of DNA polymerase gamma, the sole enzyme responsible for replicating and repairing mitochondrial DNA. We'll start with Dr. Phil West and Professor Bill Copeland to explain how a mitochondrial disease like POLG begins, how that manifests in patients, and how much more work is needed to tackle this devastating condition. Okay, Phil, could you please start off by telling me about mitochondrial malfunction and what starts to go wrong there?
So you might think of mitochondrial dysfunction if we use the analogy of running out of oil in your car. If your car engine runs out of oil, the engine starts to fail, but many other critical systems in the car also fail. And likewise, this is similar with mitochondria. As the organelles that power our cells, or the cellular energy factories, if they start to fail, often other critical aspects of the cell also fail. And these could be protein quality control, nuclear DNA maintenance and stability. Those could start to fail. The other thing is that an overheating engine will release toxic gases, and you can think about mitochondria in that way as well. Damaged mitochondria will release things, metabolites or other molecules into the cytoplasm or other parts of the cell that will lead to cellular breakdown and dysfunction. One of these could be reactive oxygen species, which are highly damaging reactive oxygen molecules that impact proteins, nucleic acids, and lipids in your cells. And so the breakdown of mitochondria in our cells really do manifest as if your car is running out of critical fluid, such as the oil or something else.
And how does that damage start on a DNA level?
Our wild-type POLG gamma makes a small amount of mutations, but when you have some of these mutant ones, and there's a lot of mutants POLG that we've characterized, depending on the severity, 2 things will happen. If the gene defect is so severe, the enzymes just don't work, and they actually can't copy the mitochondrial DNA. And so what's happening is that you get what's called depletion. And some of the early childhood diseases, they're not so much point mutations and deletions, but they're just depletion of your mitochondrial DNA. And that's very hard to recover from. The later onset ones in your adults cause point mutations and deletions, and they don't cause the depletion. And depending again about how severe it is and when it presents is really about how severe that disease will be.
Then zooming in even further, when does that damage start turning into symptoms?
Mitochondrial DNA is multi-copy number. And unlike in the nucleus, you've got 2 copies of every gene. The situation, though, is that it's multi-copy number and makes up, you can have about several thousand, from 1,000 to as much as 10,000 copies per cell. You can have a mixture of what's called wild type and a mutant form, and we call that heteroplasmy. And so if you've got like 1,000 copies in your mitochondrial DNA in a cell, and 20% has a mutation, you're likely still fine. And what has been shown in the literature is that usually you need to, in terms of the mutant heteroplasmy, has to reach well over 50% before a disease phenotype symptoms should start to show up. And so that huge copy number is a buffer per se that against some of the diseases, so they don't show up earlier.
They're actually identifying variants in mitochondrial genes at a much more rapid rate, and so I think the prevalence of these disorders is perhaps more widespread than initially thought. And so, given that there's an increasing appreciation for the prevalence of these disorders, I think it's really important for the public to understand them. And also, as I mentioned earlier, studying rare disease often provides windows into more common disorders.
How does that slow burn accumulation of mutations and DNA damage complicate diagnosis?
That the advance and certainly the improvements in technology on DNA sequencing have sort of revolutionized not only our ability to diagnose these diseases, but also just the cataloging of the large number of pathogenic variants that exist. I mean, we now know that there are over 300 pathogenic variants in the POLG1 gene that result in POLG-related diseases across the spectrum. And so I think we're getting better at not only identifying those mutants, but also sort of tying those mutations to particular POLG-related disease phenotypes.
Is the onset of disease then just a matter of chance, or could there be some underlying cause that might be isolated?
Mitochondrial themselves lack certain DNA repair processes. There's a lot of sub DNA repair processes, but there's three main ones. There's one's called mismatch repair, and so we study like the fidelity of the actual DNA polymerase, and the Paul gamma polymerase has a very high fidelity. Nonetheless, like the nuclear DNA polymerases, they occasionally will make a mistake, insert a wrong nucleotide across a template base. You've got like your A should pair with T, adenine, I guess thymine, and guanosine, called cytosine. Sometimes it makes a common mistake like putting in instead of a going to a T, it'll put in a C instead. And that occurs on even of your best DNA polymerases, Paul gamma, Paul delta, and Paul epsilon, which are your main replicative DNA polymerases. It occurs about 1 in 10 to the 5th, 10 to the 6th, about 1 in every 100 to 1 million events. The enzymes themselves have an intrinsic, what's called proofreading activity, and that's also Paul gamma has that. That adds on another level. Well, that means you can think of it as typing, and that's the best analogy. As we type, the actual fidelity of typing is what we call insertion fidelity. When we make a mistake, we hit the backspace key. That's analogous to proofreading. But after we're done, we still want to do a spell check. That's a whole different protein machinery called mismatch repair.
I'm an immunologist by training, and I can say that the field of, or the understanding of how mitochondria impact immunology has grown dramatically in the past 15 years. And so, I think about mitochondrial diseases as having an immunological component. And we know from some of the literature that patients with POLG-related disorders, especially Alpers syndrome, have this interesting relationship with viral infections. And so viral infections seem to precipitate, or can in some individuals precipitate, mitochondrial disease, specifically liver failure and seizures. And we know that there's this connection. We don't quite understand it. And I think what I'm really interested in, and collaborating with other clinicians, scientists and clinicians, is trying to understand that, and that relationship between infection and the immune system and mitochondrial disease. And I think that this is critical because if we think about childhood mitochondrial diseases, anyone with children knows that kids are often experiencing viral infections early in their life, and this could be a precipitating factor for mitochondrial disease, and also could accelerate the progression of the disease.
And how fast is research advancing to meet patients' needs?
Well, actually, almost in like the last 10 years, I think 15 years ago, someone asked me, well, what's the outlook? And I didn't have any future wisdom about these new therapeutics in the pipeline, and it didn't look that grim. Right now, it looks fantastic. There's some really good therapeutics coming online, really good options. But a good parallel case is what I like to call the case that with the TK2 patients. That is a mitochondrial thymidine kinase. And that was also for young children, it was pretty much a death sentence. Now they undergo nucleotide therapy, and it pretty much reverses almost 100% of the disease. And there's nucleotide therapy that is being used for POLG gamma diseases with mixed results, but they show some promise. But I think some of those combinations, and maybe in combination with the pretzel drug, you may be able to delay this for decades, many decades. Yeah. So the future is bright.
And then what's needed to move it faster?
Critical funding for studying and understanding rare genetic diseases often has unbelievable impacts on more common diseases. And so the funding body or the public at large sort of receives dual benefit for studying these rare diseases because they impact our understanding of common disease.
And mitochondrial diseases in general need to be better appreciated. And organizations like the POLG Foundation, UMDF, Mito Matters, and Mito Action, they're great. They're great. And we would be suffering probably without them, because the public awareness is vital.
And so funding is absolutely essential. I think the POLG Foundation has done a really wonderful job in funding some of these early studies. The United Mitochondrial Disease Foundation is also doing a great job. There are many other and a growing list of organizations that are interested in mitochondrial disease, and this is absolutely critical for advancing hopefully not only the knowledge of the disease, but also treatments, and hopefully in the future cures.
Definitely more money is needed. So whatever your funding situation, for the U.S., it's NIH, so we have to appeal to our Congress people for more funding and just in basic research. The basic research has played out as some of the most, it's a long-term investment, but it's definitely some of the best investments in terms of understanding of where to go. And the other one is just to push for a molecular diagnosis.
And Bill, to follow up on the point of molecular diagnosis, that brings us right back to the start of the patient's journey through their diagnosis, hopefully getting to the right diagnosis. How urgent is that? And how likely is it if clinicians are not thinking about POLG in the first case?
What we've seen is that patients go to their primary physician, and the primary physician isn't educated in a mitochondrial disease. So if they're lucky enough, they get referred to another physician. But we've seen patients where they literally get bounced around from doctor to doctor. And if they're lucky, they find the right specialist who will give them the right test and get the right diagnosis. But the question is, how many people never get to the right physician, to the right specialist, and never get the correct diagnosis? So that's we'd like to solve that problem as well.
This was an initial taste of POLG, and for more about the POLG Foundation, head to the episode description accompanying this episode for links, material, and support. Stay tuned to our next episode featuring Jeff McDonald, an adult POLG patient sharing the personal journey and advocating for the POLG community. Until then, goodbye.